Thank you. Well, this talk was probably accepted, our paper was probably accepted with the specific intent of scheduling it last in a session because what we've done is almost embarrassingly simple. So it could be a very short talk, but I will find some things to say. So we've been working with a specific biological collaborator who is growing prostate cancer in mice. She has a bone metastasis that she's been growing in skid mice. And she's studying, amongst many other things, gene expression. And to that end, she wants to be able to cut out a piece of the tumour, extract the RNA, and do RNA-seq. The issue that she faces is that inevitably there is a mix of cells. There will be some mouse cells and some human cells, and because the gene expression is different on a per gene basis, it's not obvious always which organism the gene is being expressed in, especially where you have homologous genes where the reads can map to either reference genome. So it turns out that all we do is something incredibly simple. She has considered FACS sorting or laser capture microdissection. We heard this morning that might actually be a good route to go. But in this case, what we're doing is we're sequencing the whole lot and we're separating the two classes of reads after. And this actually has some potential benefits because you're getting a snapshot point in time that tells you about the gene expression in the stroma, in the surrounding tissue, and the gene expression in the tumour itself. So although we haven't pursued that line of inquiry in detail, there is certainly some interesting investigation to do there about studying interactions between the stroma and the tumour. So really what we do is remarkably simple. We could do a very simple method based with top hat, and we did this for evaluation purposes, where we simply align the reads to both genomes. You just run top hat again, and then you take the SAM files and you just use a trivial Python script and you say, did this map on this side, did this map on the other side? And this approach actually works really quite well. A large fraction of the reads align specifically to one or the other. Quite a lot of the reads, there are a significant fraction of reads that do align to both. And if Top Hat produced meaningful mapping quality scores, you could perhaps do some statistics based on those scores to try and resolve some of those cases where it maps to both genomes. But that would make for too short a paper, so we didn't stop there. Instead, what we have developed is a method based on k-mers. Essentially what we do is we find all the set of k-mers in the host genome and all the set of k-mers in the graft genome, and then we just build that Venn diagram again. And each k-mer can be classed as being coming from both, or occurring in both, occurring in the graft genome, occurring in the host genome. And just to try and avoid confounding problems from sequencing errors, we also pay attention to the case where a k-mer occurs, say, in the host genome, in the mouse genome, but not in the human genome, but there is a Hamming distance 1 neighbour. Okay, so there is a near neighbour, because if there is a base calling error in the sequencing, then you might assume the wrong gene. So we collect that set as well, and we call them the marginal set. So then we have something that looks like this. Then when we get our reads, we can classify them very simply. We take each read or each pair and we decompose it into its constituent k-mers. We use that reference set that we've built. We look up the class of each of those k-mers, and we can use a very simple little decision table. If there were graft k-mers but no host k-mers, We don't really care whether some were in both. We have evidence that says that it was from the graft, so we call it that way. Likewise for the host. If there are only k-mers that occur in both, then we can't tell. We call it both. If there are no k-mers at all, because there were so many errors or from a different origin that none of the k-mers occur in the reference set, then we say that it occurred in neither. And if if you only get marginal k-mers, we call it too hard and say it could have arisen from both. So this approach is only going to work if the marginal set and the both set are small, if there is good discrimination. And in the specific case that we look at, where we've got the human genome and the mouse genome, and we've chosen k equals 25, and you can ask me after why we chose k equals 25. It was because we started there and it worked so well, we actually never changed k. That was the choice. So it turns out that there are about 2.4 billion k-mers in the human genome, about 2.1 in the mouse genome. The union is about 4.5, that is almost the full size, because the intersection, that middle both section, is out of the 4.5 billion k-mers is only 12 million, which is about 1 in 300 k-mers in the human and mouse genomes are common, which is surprising. We were kind of astonished, but it is thus. And the marginal set, actually, I was a little bit worried, conservation, you would maybe expect that there was a substantial kind of crosstalk in that marginal set, but it's only a little bit bigger. And it's about 1 in 130 k-mers, which in practice, when you take all that sort of ambiguous region, it's a little bit less than 1% of k-mers. So we did some experiments to see if this actually works, and we'd used k equals 25, human and mouse. We took a library of mouse RNA, a library of human DNA off the sequence read archive, and our third one was actually a Xenograft sample from our collaborator. Just to note on performance, on our particular machine that we have, to run top hat twice against the two genomes on the BM18, the Xenograft sample, takes about 3 days, and then a trivial amount of time to do the post-processing. Our tool using these k-mer methods takes about 2 hours, so there's quite a performance disparity, though of course in honesty it is worth noting that you actually get alignments out of top hat, you only get the classification out of Xenome, but you can then take that set of classified reads and run the human fraction with top hat on the human reference and the mouse fraction on similarly, and you'll actually take a lot less than 3 days. It'll take about 1.5 days. So on the two samples, as you'd expect, the 4 columns in each case are human, both mouse and neither. The middle grey bar shows which reads, the number of reads that both methods produced the same results. The light bar at the top shows the number of reads classified into that class only by the k-mer method, and the dark coloured region at the bottom shows how many reads are classed thus by the top hat based alignment method only. And what we see is that actually they agree substantially, though there is in each case on the mouse data and on the human data, there is a substantial number that are correctly classified by the k-mer approach that are not correctly or classified by the alignment approach. But once again, this is mostly accounted for by cases where you get a high quality mapping to the correct one. You get a mapping of low quality to the other one. So you could resolve this with mapping at some computational cost. So then we do it with our mixed sample, and sure enough, again, we get a very similar picture. It looks like this sample is roughly 80% human and roughly 20% mouse. How can we validate to see if that is actually the case? Well, we have some real-time PCR data from our collaborator. So we used the alignments and correlated, well, plotted against the QT-PCR. So we've got FPKM along the, or log FPKM along the horizontal axis, and the QT-PCR relative to a housekeeping gene. And you can see that basically they fall along a diagonal line showing that they largely agree. We spent some time investigating why that outlier on the left exists, and we came to no conclusion other than the fact that the reads just aren't there in the RNA-seq data. So there are a bunch of things that we could do better. It's currently using very simple set membership tests, so you could argue that maybe we should be doing something statistical. We're paying no attention to how many k-mers out of a read pair were classed as human, how many were classed as mouse. We just are using a very simple algorithm, so maybe that might make a difference. The marginal sets at the moment are stored together, so we don't distinguish between something that's human and looks like maybe it has a neighbour in mouse or the reciprocal. And you could once again get very slightly more accurate classification if you paid attention to that. Though the classification, as was apparent, is actually quite good, so that would be a marginal improvement. There are a number of, what I haven't talked about at all is how we actually represent the k-mers, and you can look in the paper, there's a pile of technology that we use to represent the large set of k-mers efficiently. There have been a bunch of papers just in the last few months about using Bloom filters to represent sets of k-mers, and certainly that would reduce our memory usage significantly. If you download our tool, which there's a URL on my last slide, you'll see that it runs. We've had just a couple of emails in the last day. Some people have downloaded it and got an uncaught exception, and we're pretty sure it's because their file system has filled up. You do actually need enough temporary file system and file system to store it, and we will fix that error message. In fact, Jeremy's nodding. He may well have actually put a patch on the website already. But we do hope to reduce the memory usage. It runs, it'll run on a desktop server. I think it uses 18 gigabytes of RAM for the reference structure. So it'll run on a server, and a Bloom filter would be an obvious improvement that might get it down onto your laptop. So that's really all there is to it. It's a very simple thing. It allows you to bin your reads, which you can then process further. And it remains just to acknowledge our collaborators. Particularly, I wish to acknowledge Isaac Haviv, Izzy. For complicated reasons, we failed to include him in the acknowledgements in the printed version of the paper. And you can download it. There is a URL there. And we have some related material in a poster tomorrow, U42, and tools for download. We have Xenome, this tool. We have a de novo assembler, and we have coming any day now, we have a transcriptome assembler as well. Any questions? Yeah.
So, very nice talk, thanks. Xenograft sample is usually used to identify SN mutation or fusion gene in cancer research.
Yeah.
So do you think this method can be applied to identify mutation or fusion gene?
Certainly, I mean, this is exactly the case.
Yeah.
We're dealing with prostate cancer, which of course is known for having many, or relatively many, fusion genes. It certainly does appear to be the case that it's still classifying the reads correctly. You only need one correct k-mer in the read pair in order to get the correct classification. So really what it's letting us do is just filter the data to deal with the mouse contamination, finding the fusions. You will then probably want to run Top Hat and Fusion Finder or do de novo assembly on the data. So it's kind of orthogonal.
Okay, thanks.
Actually, my question was similar in a subset of that, but just about the SNVs. Have you done any looking at calling SNVs from the results of this?
No, we haven't.
Okay.
Once again, the same kind of argument applies. As long as there is one correct k-mer in the read pair, then you'll still get the right classification. Details of things like actually mapping them is then a downstream thing. And we did this to save time.
Mm-hmm. Does it treat read pairs as a unit or as separate reads?
Yeah, you can treat them as a unit.
Okay.
Yeah.
Are there more questions?
So for your k-mer, you have the marginal read. You didn't assign to any organism, right? To human or mouse.
Sorry?
For your marginal read,
Yeah.
you didn't assign to human or
No,
mouse.
we treat them as if we don't distinguish between the origins.
Have you ever, because for the same gene, for the one gene, you have homology in human and mouse. So maybe certain regions you have unique mapping. Then you can maybe just try the EM algorithm to assign those marginal reads. Then you can able to quantify the gene, right?
Yeah,
have you ever
so
tried?
in the paper there is a diagram showing for the distribution across genes of how many distinctive k-mers there are, and it turns out that almost every gene has got distinctive k-mers.
Yes.
So it's actually quite unlikely that you'll completely fail to distinguish the mouse version from the human version. There are a couple of cases, there are a couple of microRNAs and things that are too homologous, but it's quite rare.
Yeah, I just try to see how to quantify the gene expression in xenomouse.
Yeah, so it doesn't do alignments. So to do the gene expression we actually used the Top Hat alignments on the human fraction run with Xenome. So Xenome is really just trying to separate out the human and the mouse. It's not trying to do anything else. So if you want to compute gene expression, then yeah, you need to take the human fraction or the mouse fraction and use Top Hat or cufflinks or other tool chains.
Okay, thank you.
Are there more questions?
You're saying that maybe you should be doing something statistical with the reads to say what % chance, or why, is there a threshold? I'm wondering whether you even need that. It sounds like the program already does a good job of classifying.
Right, so that's exactly why we haven't.
Yeah, I mean, it
Right,
sounds like...
that you could get you could get a marginal improvement, and it might give you a warm, fuzzy feeling that what you were doing was well founded, and you could defend it to your reviewers better. But
your time's probably better spent elsewhere.
right.
So you're saying you had the lay reviewers, which is
No, I thought we had terrific reviewers. They said this is a really obvious thing, but it solves a problem. It's like, great. You understood what our paper was about.
Are there more questions?
Hi Tom, can you change K?
Yeah, it's not hard-coded. There is a default value of 25, oddly enough.
Reduce K to reduce your memory requirement?
Yes, you could. We've only done very ad hoc trials, but if you reduce it too much, you will increase the set of k-mers that are ambiguous. So feel free to do some experiments and report to us.
No worries.
Yes. When you compare mouse to human, you look at DNA. But if you look at the transcriptomes, you look at the transcript overlap, like a transcript.
No, so we have been you could use some reference transcripts in either case. We actually used the genomic references on the basis that we're not trying to distinguish read-throughs from, finished RNAs and any of those things. So we're just looking for k-mers. In the presence of short exons, you might have some loss of k-mers, but once again...
My question is, do you see higher percentage overlap when you look at the transcript sequence alone?
You're right.
Compared to the whole genome?
Yeah. We haven't done that specific comparison, but I suspect that there is still actually very strong the results suggest that actually the separation is still very strong, but I would need to quantify that.
Yeah. So I should just say, I'm one of the developers of Xenome, Tom's colleague. There is actually a flag, a parameter to Xenome, which allows you to impose an upper limit on the amount of memory you can use. So although the entire index, which consists of all of the k-mers in the graph, will occupy 18 gigabytes in total, you can tell Xenome to use at most, say, a 4 gigabyte slice of that at a time, and it will classify your reads by default passes.
Thank you, I forgot. So you can run it on your laptop, it just will take a bit longer.
More questions? Okay, so